Let's have a moment of science and surprises of chemistry. Good afternoon to all. It's a day of pleasure to all Stephanians. Department of Chemistry, St. Stephen's College, Uzhavoor is organizing an international webinar on chemical reactions in frozen environment. It's a pleasure to extend a warm welcome to one and all to this webinar. The goal of this webinar is to update advancement in chemistry which are involved in the frozen environment. Ice is present in the atmosphere, terrestrial surface, and ocean environment. So, today we will come to know about the magical surprises of ice. I wish you a wonderful time ahead. First of all, I invite Ms. Bibi Joseph, our department faculty, to deliver the welcome speech and also to introduce our speaker, Dr. Sunil Paul Mathew M. Welcome, Miss.
Good morning to all. A respected bursar of the college, Reverend Father Jins Nellikatil, Head of the Department, Mr. Ebi Jimson, my colleague, Dr. Arun Thomas, participants from various institutions, and my dear students. We are about to start the third talk in the international webinar series organized by the Department of Chemistry, St. Stephen's College, Uzhavoor. We had overwhelming response from the participants of the first and second talk, which were delivered by Dr. Annu Thomas on the topic Chemistry and COVID-19, and Dr. Srijit Mangalat on the topic Two-Dimensional Nanomaterials for Biomedical Applications. This webinar series is motivated by responses of the various webinars organized during the pandemic period. It is indeed a matter of privilege to overcome you all to this auspicious function. Respected Father Jins Nellikatil, always instrumental is working our college into new heights. He works in rapport with the principal and other stakeholders to bring the college into work forefront, always supporting the department in all our infrastructure development projects generously. I, on behalf of all the participants, welcome you, dear Father. Resource person of the day, Dr. Sunil Paul Mathew M., hailing from a middle-class family from our state and having worked in different internationally reputed research organizations in a role model to all the young researchers. He had won his PhD at Mahatma Gandhi University, Kottayam. He had his first postdoc in chemistry at Division of Environmental Science and Engineering in Korea. Now he is working as a postdoctoral researcher at Department of Social Science and Soil Protection in Prague. Whenever we listen a lectures in chemistry, it will be about some chemical experiments that is happening between the walls of a laboratory. Now we will listen to this a talk which happens outside the laboratory. I am quite sure this will ignite the young minds to pursue their research in chemistry of nature. On behalf of all the participants, on behalf of St. Stephen's College, Uzhavoor, and with immense pleasure, I welcome you, Dr. Sunil Paul, to this webinar. Our head of the department, Ebi sir, is leading us. He is always supporting and suggests us with new innovative methods to improve our teaching and improve the learning outcomes. I cordially welcome you, sir. Also, I would like to welcome Dr. Arun Thomas, my colleague, to this webinar. We have participants from many institutions from India and abroad too. The response from the participants is always motivating us. I wholeheartedly welcome all the participants to this webinar. Our students are the ultimate objectives of this webinar. I welcome you all, my dear students. Thank you.
Thank you, ma'am. The success of our institution surely depends on its management. We proudly invite our college bursar, Father Jins Nellikatil, to inaugurate our international webinar on chemical reaction in frozen environment and also bless the occasion with your words. Welcome, Father.
Thank you. ഇന്നത്തെ ഈ വെബിനാറിന് നേതൃത്വം നൽകുന്ന ബഹുമാനപ്പെട്ട ഡോക്ടർ സുനിൽ പോൾ മാത്യു സർ, ഉഴവൂർ സെയിൻറ്റ് സ്റ്റീഫൻസ് കോളേജ് ആൻഡ് കെമിസ്ട്രി ഡിപ്പാർട്ട്മെന്റ് HOD, പ്രിയപ്പെട്ട എബി സാർ, ബിബി മിസെ, അരുൺ സാർ, ഈ സെമിനാറിൽ പങ്കെടുക്കുന്ന പ്രിയപ്പെട്ട സുഹൃത്തുക്കളെ, ഒരുപാട് സന്തോഷമുണ്ട്. ഉഴവൂർ സെയിൻറ്റ് സ്റ്റീഫൻസ് കോളേജിന്റെ ആഭിമുഖ്യത്തിൽ കെമിസ്ട്രി ഡിപ്പാർട്ട്മെന്റ് നേതൃത്വം നൽകുന്ന കെമിക്കൽ റിയാക്ഷൻസ് ഇൻ ഫ്രോസൺ എൻവയോൺമെന്റ് എന്ന വിഷയത്തെക്കുറിച്ച് ഒരു ഇന്റർനാഷണൽ വെബിനാർ സംഘടിപ്പിക്കുന്നു എന്നുള്ള കാര്യത്തിൽ കെമിസ്ട്രി പഠിപ്പിക്കുന്ന കെമിസ്ട്രി ഫീൽഡുമായിട്ട് ബന്ധപ്പെട്ട് നിൽക്കുന്ന എല്ലാവരും, നിങ്ങൾ എല്ലാവരും ഒരുപാട് ഭാഗ്യം ചെയ്തവരാണ്. കാരണം കെമിസ്ട്രി ഒരു റോയൽ സബ്ജക്റ്റാണ്. സയൻസ് സബ്ജക്റ്റുകളിൽ ഒരു റോയൽ സബ്ജക്റ്റാണ് കെമിസ്ട്രി എന്ന് പറയുന്നത്. പ്രത്യേകിച്ച് ഒരുപാട് സ്കോപ്പ്, ലോകം മുഴുവൻ ഇന്ന് കെമിസ്ട്രികളെയാണ് സൂക്ഷിച്ച് നോക്കിക്കൊണ്ടിരിക്കുന്നത്. നിങ്ങളിൽ നിന്ന് ഒരുപാട് സംഭാവനകൾ ഈ സമൂഹം മുഴുവൻ പ്രതീക്ഷിക്കുന്നുണ്ട്. പ്രത്യേകിച്ച് സമൂഹത്തിൽ ഒരുപാട് ഗ്ലോബൽ വാർമിങ്, ക്ലൈമറ്റ് ചേഞ്ചിങ്, ഒരുപാട് പ്രതികൂല സാഹചര്യങ്ങൾ സമൂഹം നേരിട്ടുകൊണ്ടിരിക്കുമ്പോൾ അതിനൊക്കെ സൊല്യൂഷൻസ് കണ്ടെത്താൻ സാധിക്കുന്നത് കെമിസ്ട്രി ഫീൽഡുമായിട്ട് ബന്ധപ്പെട്ട് നിൽക്കുന്ന നിങ്ങൾക്കാണ്. നിങ്ങൾക്ക് ഒരുപാട് നല്ല സംഭാവനകൾ സമൂഹത്തിന് നൽകാൻ സാധിക്കും. പ്രത്യേകിച്ച് കെമിസ്ട്രി പഠിപ്പിക്കുന്നത് കൊണ്ട് എന്തെങ്കിലുമൊക്കെ പ്രയോജനം സമൂഹത്തിന് നൽകാൻ സാധിക്കണം എന്നൊരു ബോധ്യത്തോടു കൂടി ആവണം ഈ സബ്ജക്റ്റിനെ നിങ്ങൾ സ്നേഹിക്കുന്നതും ഈ സബ്ജക്റ്റിനെ നിങ്ങൾ പഠിക്കുന്നതും. തീർച്ചയായിട്ടും ഈ ഒരു സെമിനാർ ഓർഗനൈസ് ചെയ്ത കെമിസ്ട്രി ഡിപ്പാർട്ട്മെന്റിന് എല്ലാവിധ അഭിനന്ദനങ്ങളും നൽകുകയാണ്. അതിന് നേതൃത്വം നൽകുന്ന എബി സാറിനും ബിബി മിസ്സിനും അരുൺ സാറിനും ഒപ്പം കെമിസ്ട്രി ഡിപ്പാർട്ട്മെന്റിലെ സ്റ്റുഡൻസിനും ഈ സെമിനാറിൽ പങ്കെടുക്കുന്ന നിങ്ങൾക്ക് എല്ലാവർക്കും എല്ലാവിധ ആശംസകളും നേരുന്നു. വളരെ പ്രത്യേകമായിട്ട് ഈ സെമിനാറിന് നേതൃത്വം നൽകുന്ന ഡോക്ടർ സുനിൽ പോൾ മാത്യു സാറിന് എല്ലാവിധ ആശംസകളും നേരുന്നു. നല്ല ക്രിയേറ്റീവായിട്ടുള്ള ക്രിയാത്മകമായ ചിന്തകൾ പങ്കുവയ്ക്കുന്ന ഒരു മണിക്കൂർ സമയം നിങ്ങൾക്കുണ്ടാകട്ടെ എന്ന് ആശംസിക്കുകയാണ്. നിങ്ങളുടെ എല്ലാവരുടെയും പേരിൽ ഈ വെബിനാർ ഔദ്യോഗികമായി ഉദ്ഘാടനം ചെയ്തതായിട്ട് പ്രഖ്യാപിച്ചുകൊള്ളുന്നു. എല്ലാവിധ നന്മകളും എല്ലാവർക്കും ആശംസിക്കുന്നു. താങ്ക് യു.
Thank you, Father. Webinar platform is set. The little scientists are ready for brainstorming. Even the climate and environment is ready to go with the flow. I would like to welcome our honorable chief guest, Dr. Sunil Paul Mathew. Sir, we are so grateful for your presence and also to get the ideas from you about chemical reactions in frozen environment. It's time to hear from the resource. Welcome, sir.
Thank you very much. So, my sound is clear. Is everything fine?
Yes, sir. It's clear.
Okay, great. So, good afternoon, everybody. First of all, thank you very much to the Department of Chemistry, St. Stephen's College, Uzhavoor, for the kind invitation. Also, a special thanks to Bibi Joseph for the introduction, as well as, of course, it is a little bit exaggerated, but I really like it. But there are some corrections. First of all, I am not working in Department of Social Science. It's actually Department of Soil Science and Soil Protection. Anyway, it's fine. So I hope all of you are fine and most importantly safe in the present situation. Today, we will discuss about chemical reactions in frozen environment, or simply ice chemistry. First of all, I am almost sure that most of you are not much aware about this topic. So I will try to give you some very basic but not very detailed introduction about this topic. Then we shall discuss some of our experimental results. So before going to the talk, as an introductory note, I would like to mention few points. First of all, this is my very first online lecture. So in any case you feel some issues, for example, my sound is not clear or something like that, please feel free to interrupt my talk. Okay. Then secondly, and most importantly, I should mention that I am not working in this topic anymore due to various reasons. It's mainly because I recently changed my position into a different university and the priorities are changed, topic is changed, like that. Anyway, I did this work between 2015 to 2019 when I was a postdoctoral researcher at Pohang University of Science and Technology, also known as PostTech in Korea, not the Kim Jong-un's North Korea, it's in South Korea. At that time, I was working with this person. His name is Wan Yong Choi. He is a professor in the Environmental Engineering Department of PostTech. If you are someone who is interested in like topics like photocatalysis or water splitting or environmental remediation, you probably hear his name before. As you can clearly see from his area of expertise, its ice chemistry is not there. That is because ice chemistry is not his first priority research area, not because it is a bad topic, but due to various other reasons, for example, funding, etc. Also, neither me nor Professor Wan Yong are experts in this area. So our strategy is more like trying something new and learning some new things. So you will probably wonder why I am telling all these stories in advance. It is because in my slides, you will see many things that are not that much completely elucidated or completely known or still under investigation. For example, many of the mechanistic aspects you are about to see in my slides are still under investigation or not completely known. It's not because they are wrong, it's because it's because it's still under investigation by the scientific community. Okay. So, why ice chemistry? As we all know, most of the chemical reactions are temperature dependent or even temperature sensitive. For example, when we increase the temperature, the reaction rate generally increase, or when we decrease the reaction temperature, usually the reaction rate decreases. Okay, of course, there will be exceptions, but we are not considering that today. Now I have a question to all of you. How many of you ever thought about the situation once the temperature is below zero? That is, the reaction medium is completely frozen. So, this is one of the first report in this area, which really motivates many researchers like us. In the beginning of 1990, a Japanese scientist, his name is Takanaka, who interestingly found that the oxidation reaction of nitrite to nitrate, which is a really slow process in the normal situation, that is in the aquatic environment, is exceptionally fast in frozen conditions. That is around 10 raised to 5 times faster when the reaction conducted in frozen environment. So this is a really interesting observation. It really motivates many researchers around the globe, including our laboratory. So these are some other examples. The first case is the photolysis of 2-chlorophenol, and the second one is the photolysis of 4-chlorophenol. In both cases, as you can see in the aquatic environment, the main products are dihydroxy compounds such as catechol or hydroquinone. However, when the same reaction is repeated in a frozen environment, some dimeric products are formed. For example, chlorodiphenyl, phenol, etc. These products are actually much more toxic to the environment, as well as much more persistent in the environment. So it become a really important environmental issue. Okay. So now I will show some more examples about the to show the importance of this topic. I'm not going to explain all very deeply. The first case is actually the protonation of cresol red. It's a widely used dye in industry. The authors of this work found that actually it's Journal of Physical Chemistry. I missed the first J. Anyway, the author of this articles found that the protonation of cresol red is exceptionally fast in frozen state than its aqueous counterpart. That is approximately 2 to 4 order of magnitude fast, depending on various factors such as the freezing rate, presence of other solutes in the medium, etc. The second example is more like a statistical study, and it is about the enhanced aggregation of another dye called methylene blue. So the authors found that the aggregation is really fast in frozen environment than in aqueous environment. So this is another interesting result published in the last year. It's about the synthesis of a two-dimensional polymer, actually in the ice surface. This is the title of the article. The authors behind this work recognized that the ice surface allows a very rapid polymerization of the monomers in a much efficient manner than in the normal condition, that is in the aqueous phase. So again, I am not discussing this work more. Rather, I will move to the next slide and try to explain some of the results published from our laboratory. So these results are actually published by my senior colleagues. The first one is about the photoreductive dissolution of iron in presence of, say, some small organic ligands. More specifically, in this case, the effect of formic acid is considered. So as you can clearly see from this figure, the reductive dissolution of iron is clearly enhanced in frozen solution, that is here, whereas the same in normal aqueous phase is very low or even negligible. Also, you can clearly see from this figure that the dissolution trend is increased when the concentration of formic acid is increased. That is again only in the frozen condition. So let's check the second figure. It's actually about the dark phase dissolution of iron. and the red color represents the frozen samples, and the black color represents aqueous samples. Again, as you can clearly see from this figure, the dissolution of iron is actually non-reductive dissolution. So this is total iron. So you can clearly see from this figure that the non-reductive dissolution of iron is clearly enhanced in presence of a number of organic, simple organic molecules. For example, formic acid, acetic acid, etc. Also in presence of some complex molecules like humic acid, fulvic acid, etc. So at that time, when this work published, the authors suggested that this is a new pathway for the generation of bioavailable iron in the environment, especially applicable to the frozen environment. This is another important result published from our laboratory in 2017. It is about the reductive removal of bromates. Bromate is a very toxic disinfectant byproduct, which is usually formed during ozonation. Ozonation is an important water purification method widely used throughout the world. So bromate is significantly toxic compound. In normal situations, this bromate will slowly reduce to bromide, and thereby its toxicity become negligible or removed. However, our laboratory members find out that this slow reduction process is extremely fast in frozen conditions. For example, these are some of the quantitative aspects. In dark condition, nearly 30 to 40% of bromide is converted within 1 hour, within 1 day. However, in the case of UV irradiated condition, up to 90% bromide will be removed, whereas the slow reduction rate of bromide to bromide in normal aqueous phase is like less than 1% in 1 day. So this is a really interesting result. These kind of results revealed that the reaction rates and sometimes the reaction mechanisms that are well known in aqueous solution may not be completely applicable to frozen conditions. So some of them, for example, some of the examples even induces an unexpected toxic situation in frozen condition. That is our main motivation to work in ice chemistry. Now, the important question is how these kind of differences happens in frozen environment. Most of the authors reported their results on the basis of either freeze concentration effect or another parameter called freezing potential. So in this case, I will first explain what is freeze concentration effect. Instead of explaining this definition, I will try to explain the same with the help of this figure. Basically, this figure is an optical image of ice. As I hope you clearly distinguish two separate portions. The one is a relatively big portion called like a more crystalline solid part. This is the majority of the ice. This part is called ice grain boundary or bulk ice. You can also see an interface region between two big ice crystal lattices. The first part is ice crystal lattice, not grain boundary, ice crystal lattice. In between two ice crystal lattice, you will see some small portions. According to the studies, these small portions still maintain some properties of liquids. So we can consider this portion as a liquid-like region. Also, we will call this region as grain boundary region or ice surface. So please familiarize or notice all these technical terms. We will use that for the remaining talk. That is grain boundary or ice surface, and this is the ice crystal lattice or bulk ice. Okay. So what happens upon freezing? I will focus this part. So this is the blue part is the ice crystal and the white part is the grain boundary. So what happens upon freezing is some solutes will be likely excluded from the crystal the relatively big ice crystal lattice, and as a result, they will be concentrated in the relatively small grain boundary region or the ice surface. So what happens? Since the practical concentration of the reactants are very much higher or several times higher in the grain boundary region, because of this freeze concentration effect, the reaction rate becomes very high. The second important concept is freezing potential. By definition, it is the electric charge separation occurs during freezing of slightly ionized water. More clearly, during freezing, it is very likely that some of the inorganic ions or any other ionic species are likely separated from its counter ion due to the above mentioned process. In that case, a measurable potential difference will be generated within the system. So this is anyway a much more complicated case. Now, let's go to the first case and we will try to analyze what exactly happened in the case of the nitrite to nitrite oxidation reported by Takanaka and co-workers. According to their initial results, the conversion rate of nitrite to nitrite is very low in aqueous phase. For example, with an initial concentration of 10 millimolar nitrite and 1.2 millimolar oxygen at 27 degrees centigrade, the conversion rate of nitrite is only 5% after 1 hour. However, when the reaction is repeated at a frozen condition, even with a very small concentration of reactants, for example, 0.1 millimolar nitrite and 0.25 millimolar nitrite, at minus 21 degrees centigrade, the conversion rate of nitrite is approximately 70% at 20 minutes. That's why this exceptionally high reaction kinetics occurs. However, the authors realized that this kind of really exceptional reaction rate may not be justified only on the basis of freeze concentration effect. Hence, they conducted additional theoretical studies as well as several other experiments. For example, they tried the effect of freezing rate, effect of several other inorganic species. And on the basis of their additional results, they realized that there are two main factors that are responsible for this exceptional reaction rate. The first one is, of course, the freeze concentration effect, which leads up to 2.4 into 10 raised to 3 times higher concentration of nitrite in the grain boundary region. That is what they estimated from their studies as well as theoretical calculations. And also, there is one more factor, that is a pH reduction or pH loss in the grain boundary region. It is happened because of the selective incorporation of protons into the grain boundary region. So what will happen is, due to this pH reduction, the nitrite become converted into nitrous acid, which is a more reactive species. Okay, so I seriously hope now you got some basics of the ice chemistry. Now let's discuss some of our results. For the remaining part of talk, I will mainly discuss our studies mostly on the reductive dissolution of iron. For that, I will mainly consider two articles. The first one is the ligand-specific dissolution of iron oxides in frozen solution, and the second one is a simultaneous production of iron as well as the reactive iodine species in ice. Both are published within the last 2 years. So, why iron? Okay, we all know iron is one of the most abundant element on Earth, which is extensively utilized for industrial applications from ancient times. Moreover, this element plays many important roles in biological systems, in human body or any other living species. You will often meet many compounds containing iron, many biomolecules such as hemoglobin, myoglobin, etc. In fact, without iron containing biomolecules, probably there is no oxygen transport or no electron transport or no nitrogen fixation, etc., or at least in the current known form. However, never think that iron is a perfect gentleman or gentlewoman. It is because iron is also capable to produce highly oxidizing free radicals, for example, hydroxyl radical, by Fenton's reaction or Haber-Weiss reaction, etc. As we all know, these kind of free radicals are one of the reasons for aging, DNA mutation, some deadly diseases like cancer, etc. Anyway, at the same time, using the same oxidation power of free radicals, many researchers, including our colleagues, utilized iron for environmental remediation applications. So that is also an importance of iron. Now, we will discuss about the bioavailability of iron, which is an important concept that decides the availability of this metal or basically any nutrient in the living system. By definition, bioavailability of any nutrient can be defined as the proportion of the administered substance capable of being absorbed and available for use or storage. It is well known that some substances are known to enhance the bioavailability of iron, for example, ascorbic acid, also known as vitamin C, and there are some species or some substrates that are non-inhibitors of iron, for example, some polyphenols or some metals like calcium, etc. Anyway, for a living system, it is really important to maintain the amount of bioavailable iron in an optimum form, mainly to avoid diseases related with iron deficiency. At the same time, iron overload also leads to various health issues. So let's discuss something more about bioavailability of iron. In general, our body mainly deals with two different forms of iron, that is hem and non-hem form. I'm not sure I am pronouncing correct these terms. Anyway, the hem form of iron is mainly from animal origin, that is from meat, then dairy products, seafoods, etc. They are often attached with a hem protein. Also, iron can be more easily absorbed from this hem form of iron. Conversely, the non-hem is mainly from plant origin, for example, whole grains, nuts, etc. Also, they are the absorption is typically low from non-hem iron. This less absorption of iron is mainly because the non-hem form is mainly because of the less solubility of most of the Fe3 compounds. As a result, the uptake of non-hem ion by living organisms either requires the conversion of Fe3 species into more soluble Fe2 species or ferrous ion, or the conversion into a more soluble Fe3 organic complex. So the same strategy is used in most of the iron supplements. For example, they are mainly ferrous succinate or ferrous sulfate, etc. They are all iron 2 or ferrous. Now, these are some of the reports available in the literature about iron bioavailability and the effect of various substrates like ascorbic acid. Some examples are here. Don't worry, anyway, I'm not planning to discuss all these things because they are more bio-related work and definitely beyond my scope. However, essentially, they are all reports the capability of specific molecules like vitamin C or ascorbic acids to increase the bioavailability of iron in the environment. These reports are, however, much more relevant to us. Both of these articles reports the deficiency of bioavailability of iron in polar regions, for example, Arctic Ocean. The authors of this work found that the growth of phytoplanktons are actually suppressed due to the lack of iron. They acquire these kind of data by monitoring the phytoplankton growth as well as the consumption of essential nutrients like phosphate, nitrate, etc. The authors found that the growth of phytoplankton as well as the consumption of nutrients are really exceptionally increased once they introduce iron into the medium. Okay, so these are the objectives of our studies. We mainly like to investigate the dissolution of, mainly the reductive dissolution of various iron oxides in frozen environments. For our studies, we mainly consider the effect of a number of organic substrates. We will explain more. Also, we consider some inorganic substrates, as well as we also investigated the effect of different parameters such as pH, concentration of ligands, etc. Moreover, not only the laboratory scale experiments, we are equally interested to conduct the experiments in a more realistic natural environment as well. So this is basically our methodology, which is pretty simple. Most of our experiments can be easily repeated, provided you have a deep freezer in your laboratory, as well as you have a UV visible spectrophotometer. Basically, we will mix the iron oxide samples with the ligands one by one. Then we adjust the pH using one of the acid or base here. Then we split the samples into two equal portions. One set we kept in aqueous phase and normal temperature, either in a dark or under irradiated condition like UV irradiated or solar irradiated condition, depends on the experimental requirement. The second set we kept in ethanol bath maintained at -20 degrees centigrade, again in dark or light irradiated conditions. So if you ask me why we use ethanol bath instead of other solvent baths, it is because the freezing point of ethanol is too low so that it won't be frozen at -20 degrees centigrade. That is our experimental pH. Then we take all the samples, I mean one by one at different time intervals. Then we allow the frozen samples to melt on a water bath maintained at a very low temperature. That process is called thawing. Then we filter both samples to remove any undissolved iron left using a syringe filter, and this is the specification of the syringe filter. It's actually 0.45 micron. Finally, we analyze the samples using a UV visible spectrophotometer using 110 phenanthroid method. So please note that this method only detects ferrous ion. However, some cases we really need to monitor total ion. That definitely includes ferrous as well as ferric ion. In that case, we will add a little amount of reducing agent, usually hydroxylamine, just before the analysis. I mean, after all the experimental stage, we add little hydroxylamine to the solution so that all the Fe3 become Fe2. Then we will be able to detect it. So we use this 110 phenanthroid method. Okay, these are some of the initial results. We tested the iron dissolution from goethite. It is a widely used, very important mineral of iron. So we tested a number of inorganic ions and, the effect of a number of inorganic and organic ligands. So this is some of the rules of in my slides. If the figure is black and white, the gray color always represents the result from aqueous phase as well, but the black color always represents the results from frozen case. However, if the figure is color, usually the red color represents the results from frozen condition, whereas the black color represents the results from aqueous condition. So please keep that in mind. As you can see from this figure, the results are a little bit complicated. For example, in the case of halide ions like chloride and bromide, the dissolution is almost not affected. However, in the case of iodide or hydroxylamine, the dissolution in frozen condition is significantly enhanced, whereas in the case of organic species like ascorbic acid or oxalic acid, the reduction is clearly suppressed in ice phase. Of course, the aqueous phase dissolution is very high. However, the dissolution is clearly suppressed in ice phase. So, please note that the y-axis is total ion, that is Fe2 plus and Fe3 plus. Moreover, the concentration of, the pH of this solution is 3, as well as the reaction time is 1 day. Then we monitor the speciation of dissolved ion using the above mentioned 110 phenanthroyl method, and we found that both ascorbic acid and hydroxylamine induces an exclusive reductive dissolution. It is definitely not a surprising factor to us because we know that both these molecules are highly reducing in nature. Also, the reductive dissolution of observed in the case of iodide is also good. That is nearly 90%. For all other cases, the reductive pathway is insignificant. They are all like less than 20. So our interest is more on reductive dissolution, and we will consider the first 3 cases for our further studies. Okay. In the case of iodide, we also monitor the generation of reactive iodide species such as triiodide ions. They are also environmentally relevant due to their involvement in processes like ozone depletion, mercury depletion, etc. Again, we use a simple spectrophotometric method to monitor triiodide. So you can clearly see from the left side of the figure, along with the enhanced dissolution of ferrous iron in frozen condition, the triiodide ion generation is also enhanced in ice phase, that is here. However, both the aqueous phase reductive dissolution of iron as well as the generation of triiodide is really small or negligible. Also, the table in the right side shows some quantitative aspects about the same. In the case of different iron oxide minerals, for example, hematite, magnetite, goethite, etc. The first 2 columns represents the extent of iron produced in water and ice, whereas the last 2 columns represents the extent of triiodide ions generated in water as well as in frozen condition. Of course, we can achieve a similar amount of iron dissolution much more efficiently by introducing light into the medium, into the system. For example, a significant amount of iron is produced as well as a significant amount of triiodide ion is generated when we irradiated the solution, and we achieved these results within 6 hours. So how many of you noticed the time scale of our dark phase reactions? It's actually 96 hours. That is almost 4 days. However, we achieve the same results within 6 hours by introducing light into this solution, into this system. Now, we have to verify the trend, dissolution trend. We have to really confirm it is valid. So what we did is we conducted similar studies on different iron oxide minerals and then compared the result to each other. In the first figure, we considered a number of iron oxide minerals, for example, goethite, magnetite, etc. According to these results, we found that in frozen conditions, the dissolution of iron is enhanced in the presence of iodide, irrespective of the iron oxide mineral. That is, this case, iodide always enhance the dissolution. However, in the case of ascorbic acid, the dissolution is continuously suppressed irrespective of the type of iron oxide. However, it is also clearly understandable that the extent of dissolution is definitely enhanced when the surface area of the hydroxide species increases. That is, the goethite, which has the highest surface area, yields the highest amount of iron in both cases. However, hematite gives the lowest amount of dissolved iron. In the right side of the figure, the story is almost same. However, the parameter under consideration is the ligand concentration. The first one is the ascorbic acid concentration. Here you can see this is the aqueous phase and this is the frozen phase. You can clearly see, this is the aqueous phase and this is the frozen phase. You can clearly see the dissolution is suppressed in aqueous phase, frozen phase. However, in the case of iodide, upon increasing the concentration of iodide, the dissolution in frozen phase is largely enhanced. However, in the case of aqueous phase, it is very low or even negligible. Now, this is much more interesting results, that is the pH-dependent studies. Let's consider the first case, that is absorbic acid case first. When we increase the pH, the difference between the aqueous and frozen solutions becomes less prominent. That is, in the first case at pH 3, the suppression in the dissolution is very high at ice phase. However, when we increase the pH, this difference becomes less prominent, again less prominent, and finally it's almost same or slightly enhanced. However, in the case of hydroxylamine, the situation is totally different. That is, in pH 3, hydroxylamine shows an enhanced dissolution. When the pH increases, the trend slightly changed. It's actually reversed at pH 4 and it shows a suppressed dissolution. However, in the case of iodide, there is no such trend observed. That is, freezing always enhances the dissolution in presence of iodide, irrespective of the fact that the upon pH increase, the dissolution in both aqueous and frozen solutions becomes reduced due to various other factors. So, what are the possible reasons for these kind of changes? According to our assumption, the organic molecules that are ionized in a given pH enhances the frozen solution. Why? Because we strongly believe that this kind of changes are largely depends on the pH dependent changes of this molecule. For example, ascorbic acid, which exists as neutral form in low pH. However, according to this pKa value, upon pH increases, this molecule becomes dissociated. It's deprotonated from one of this hydroxyl group. Then it becomes ionic, and we believe that once it is ionic, it start to increase the dissolution in frozen condition. In the case of hydroxylamine, the situation is totally different. That is, at pH 3, hydroxylamine is ionized. That is, it's in its protonated form. However, when pH increases, it becomes neutral. So we hope that is the case. That is, according to our assumption, the molecules that are ionized in a given pH enhances the dissolution in frozen condition. Then the next question is how. That we will try to explain in the next few slides. So this is a general mechanism. There are 3 different mechanisms of iron dissolution that explains iron dissolution. The first one is protonation, that is here. Then the second one is reduction here, and the third one is a complexation with a suitable ligand that forms a ferric complex that is soluble to water. So anyway, our interest is mainly the reductive dissolutions. So the first 2 are essentially reductive processes. Also, we can further classify the reductive process into 3 steps. The first step is the attachment of ligands to the ferric ion surface. In the second step, the ferric ion, the ligand attached to the ferric ion donates 1 electron to the ion, and it becomes a ferrous ion species, ferrous ion ligand species complex. And in the third step, the ferrous ion ligand species detached from the ferric ion compound, ferric ion surface, and it becomes dissolved into the media. So this is the general mechanism. Now let's see what happened in ice. So all these mechanisms will be really efficient in ice according to freeze concentration effect if and only if all the necessary reactive species are concentrated in the reactive grain boundary region or the ice surface. For example, consider the case of protonated hydroxylamine or iodide. We can represent all these species as X. In that case, we can see that iodide and hydroxylamine enhances the dissolution in freezing at pH 3. It must be due to the freeze concentration of all these reactive species within the grain boundary region. As a result, practically a higher concentration experiences in that region and the reaction rate increases. However, the dissolution trend is opposite in the case of compounds like neutral ascorbic acid. It should be due to the exclusion of such molecules from the grain boundary region. I represent ascorbic acid as AA, and we believe that ascorbic acids are actually trapped in the ice grain boundary. As a result, ascorbic acids are not available to react with iron and protons that are actually concentrated in the grain boundary region, which definitely reduces the reaction rate. Okay. However, an increase in pH results ionization of ascorbic acid according to this pKa value, and as a result, this molecule becomes more and more incorporated into the reactive grain boundary region. Okay, in that case, an increasing pH will definitely enhance the dissolution, as we clearly seen from our experimental results. The situation is almost same. The story is almost same in the case of hydroxylamine, however, opposite in nature. So, what do you guys think about our assumption? Anyway, when we try to publish this work, the reviewers won't fully agree with us. So, we have to do some additional experiments. What we did is, we contacted this person. I still have difficulty to pronounce his name. Probably you can try. Anyway, he is a very young but an emerging theoretician from Korea. He made some calculations and provided the following results. Please don't ask more about these results because I really don't know. These are purely theoretical results. However, as I understand or what he told to us, he theoretically predicted the binding energies of protonated and neutral forms of hydroxylamine, as well as the iodide ions in bulk ice, as well as the ice surface. According to the results, the protonated hydroxylamine can be more incorporated into the reactive grain boundary or the ice surface where the reaction happens, whereas the neutral form of that molecule prefer to stay in the ice crystal lattice. So, which clearly supports our assumption. The temperature-dependent studies also give some crucial insights to support our theory. For example, as you can clearly see from these two cases, that is iodide and hydroxylamine, when the temperature decreases, the amount of dissolved ferrous ion also decreases until the temperature becomes 0. However, once the temperature is below 0, it shows a sharp increase in the amount of dissolved ion. Okay. However, this sharp increase is definitely because of the involvement of hydroxylamine as well as iodide in the freeze concentration effect. However, in the case of ascorbic acid, although this line is not clearly linear or not really linear, it is well clear that the dissolved ion observed in this case still show a decreasing trend even after the temperature becomes less than 0. That is, the solution is frozen. It clearly means ascorbic acid is not much involved in the freeze concentration effect. In other words, the involvement of ascorbic acid is very limited in the freeze concentration effect. Okay. Finally, we designed some special experiments as an attempt to directly measure the ligands that are trapped in the ice crystal lattice. For that, we prepared frozen samples, frozen inoculate samples with ligands. So the situation is like this. You will see some red dots that represents the molecules that are trapped in the ice crystal lattice, and these yellow dots that represents the molecules that are concentrated in the grain boundary region. Okay. Now, what we did is, once the sample is completely frozen, we immediately take the samples and we crush that samples in a laboratory mortar very quickly. Then we wash the samples with cold water. We select cold water to avoid the melting of the remaining ice crystals as much as possible. Then we immediately remove the liquid part using filtration and we recollected the ice crystals. By doing so, we assume that the liquid-like grain boundary region, which is less stable, this process only wash out the liquid-like grain boundary region, and the ice crystal lattices almost remain intact. Okay, so we believe that after this washing, we will collect only the ice crystal particles, ice crystal parts. Okay, so we allow the ice crystals to melt, and we analyze these samples using ion chromatography to find the amount of species, the amount of ligands trapped in the ice crystal lattice. These are the results, and we found that the proportion of, more than 50% of ascorbic acid is actually trapped in the ice crystal lattice, whereas the amount of iodide ions trapped in the ice crystal lattice is very low. So it clearly supports our experimental assumption. Now, these are some of the experiments, some of the results obtained from real environment. In this case, we conduct experiments in an outdoor experiment in Pohang during the winter day, and the temperature is nearly 0. We actually conduct this experiment on the top of our environmental engineering building. And of course, we use some additional setups to ensure that the samples are frozen. According to these results, as you can see from this red line, the amount of dissolved ferrous ion is increased upon increasing time. Also, the amount of triiodide ions generated are also increased. And this blue line represents the intensity of solar radiation that we measure using actinometry. And please note that the amount of iron formed in the real environment, that is in the aqueous phase, is very low or even negligible. Also, these are the coordinates, geometrical coordinates of the location. Later, my colleague Dr. Kim visited King Sejong Station with our samples. This is a location of King Sejong Station in Antarctica. It is a polar research center located in Antarctica under the Korean government. So as you probably know, our Indian government also has a polar research station in Antarctica. It is actually here, and its name is Dakshin Gangotri Station. Anyway, our result from the Antarctican real environment also showed an enhanced resolution of iron in the frozen environment, that is from a variety of iron oxide, for example, magnetite, goethite, etc. Also from Arizona test dust. Arizona test dust is a commercially available sand, which is actually rich with many minerals, including iron. So we also monitor the triiodide ion formation from the same real environment as well as the iron dissolution. So both are increasing. So what we learned from our studies, we learned that the dissolution of ice in frozen environment is closely related with the nature of ligand. More clearly, freezing enhances the dissolution in presence of ionic ligands. For example, iodide and ionic hydroxylamine. However, the freezing suppresses the dissolution of iron in presence of neutral ligands such as ascorbic acid. So these are the more clear exact situation. Finally, what are the importance of these kind of results? These kind of studies are really important, mainly because similar effects were likely occurred in polar regions or soil covered under snow or ice, or in any other regions which is existed in frozen state. More clearly, depending on the nature and availability of ligands, that is ascorbic acid or iodide or whatever, a iron deficiency or a better iron bioavailability will be likely occurring frozen environment. So that is the importance of this kind of studies. Now I will show you some other examples, some other areas in which we paid attention. One of them is the reductive dissolution of lead. As we all know, lead is a toxic pollutant, toxic heavy metal, and this figure shows how lead is introduced into the aquatic environment as well as some of the symptoms of lead poisoning. We all know that lead has two main oxidation states. One is lead 2 and the other one is lead 4. It is very important, that is lead 2 is the primary species, or the lead reductive form of lead is the primary species responsible for lead pollution. Because of that, the reduction of Pb4 species into Pb2 species plays a key role in lead poisoning. According to our studies, we realized that this reduction process, that is lead 4 to lead 2 conversion, is extremely fast in frozen environment, especially in presence of halide ions, for example, bromide iodide, etc. And most importantly, in presence of a mixed halide case, the more amount of lead is formed in frozen environment. So it is definitely an environmental issue. This is another example, which is somewhat similar story, but here the oxidation of chromium 3 to hexavalent chromium is monitored. In this case, chromium 3 is considered as a relatively essential nutrient for humans, and it is not toxic and it's less water soluble. However, chromium 6 is acute toxic and it is carcinogenic. Also, it is a strong oxidizing agent. Also, it causes damages to kidneys, liver, and many other issues in our body. According to our studies, we realized that the oxidative dissolution of chromium is extremely fast in frozen environment, especially in presence of other ions like Fe3. However, these kind of processes are really slow in normal aquatic environment. So this is another environmental issue. Here is the last example where we found out that the generation of hydrogen peroxide from hydroquinone and Cu2 species is extremely fast in frozen environments. Also, the same thing has been observed for a variety of pH, and these are some of the details about mechanism, how these things happen, etc. So finally, I would like to acknowledge the following people: my supervisor and other colleagues for their timely help and support, and also Korea Polar Research Institute for the financial support. And finally, thank you very much, all of you, for the kind attention. Thank you.
Thank you, sir. Now the platform is open for questions and discussions. The speaker is ready to answer you. You can unmute your mic and ask your questions, or else you can type your questions in the chat box. So please utilize the opportunity. So there is a question from Rindu Anthony. What is mercury depletion and what are its effects?
It's quite a complicated issue. Anyway, I am not that much expert in that area, but it's very important issue in polar region. Okay, applications. I think it's a more suitable term is implication, because it will happen in the real environment. For example, all these things like the heavy metal dissolution, iron dissolution, unenhanced bioavailability, all these things will be likely happens in the real environment. Definitely, cooling requires more energy, but our intention is to know, our intention is to learn all these things happens. What are the things happens when the situation is frozen? I mean, naturally frozen, not artificially frozen. Okay, so all these things will be likely happens in the polar regions where you don't have to pay extra money for freezing. Okay.
So there is a question from A.B. Jimson. Are the conditions same in Arctic and Antarctic regions?
You mean the condition with our experimental laboratory samples, or you mean the condition in Arctic and Antarctic? I don't clearly understand the question.
I was actually asking about, because the temperature ranges vary from Arctic and Antarctic zone, and also the rate of pollution may be higher in Arctic since it is closer to human settlements. So, if you're considering heavy metal or mercury pollution like that, is there any difference in the rate of reactions like that?
There will be some difference. According to our studies, the freezing rate is also an important factor. For example, if the temperature is like very low, the reaction becomes slightly different. It is mainly because when the freezing is too fast, the molecules that are incorporated in the grain boundary or trapped in the ice crystal lattice become slightly different. So there will be some difference, but it is I don't think it's totally different. The trend will be definitely same. That is what we understand from our results.
And so, what is there any, for example, if we're considering the Arctic region, there may be some seasons where the Arctic is ice-free, some regions of the Arctic. So, when these trapped elements are released back, is there any change in the properties or any other phenomenon like that?
I don't think so. I don't think so.
Okay, sir. Thank you.
Unless there is some chemical reactions that leads to some serious structural changes, I don't think so.
So there is a question from Arya. Ligand-specific dissociation of iron oxide can occur in absence of light. Light can any effects on this reaction?
Yeah, definitely. I think I already showed that in one of my slides. Even without any ligands in presence of light, the dissolution is slightly enhanced in frozen condition. So definitely light has some effect, because the photoreductive dissolution is an important process.
Hello, Sunil.
Hi Sijo.
Happy to see you.
Yeah, me too.
So Sir, there is a question from Hari Krishnan. Effective of pH values in frozen condition different. What are the reasons behind this?
Can you show the question once again? I didn't get it clearly.
Effect of pH values in frozen condition different. What are the reasons behind this?
That is what, according to our assumption, it is because of the structural changes. I mean, when pH changes, some of our organic molecules become structurally different. For example, ascorbic acid, it's neutral in pH 3, however, it becomes ionic due to the pKa, the dissociation. So when the compounds are structurally changed, definitely the situation will be different.
Thank you, sir. Any more questions? I hope no more questions. So you got the answers of all your questions. Oh, sir. Sir, there is a question from Steffi Sunny. Can we control the concentration of reactants in the given boundaries?
The only way is to adjust the initial concentration so that it will be different. Other than that, I don't know. Anyway, it's a relatively new topic. Currently, there are not that much techniques to directly monitor how much ligands are concentrated in the grain boundary region or how much ligands are trapped in the ice crystal lattice. Of course, nowadays there are some Raman spectroscopic techniques are available. However, it's very difficult. So I think at this moment, the best way is to use the trial and error method. Like when the concentration is high, the initial concentration is high, probably the amount in grain boundary will also high. I think that is the best solution available at this moment.
Thank you, sir. Any more questions? Okay, thank you. So it's time to say a word of thanks to our chief guest. So I invite Dr. Arun Thomas, the Chemistry Association in charge, to deliver vote of thanks.
Good afternoon, all. Respected College Bursar, Reverend Father Jins Nelikatil, and our Principal, Professor Lallikis Sriyak, and the HOD of the Head of the Department, Mr. Ebe Jimson, and my colleague, Ms. Bibi Joseph, and our Chief Guest, Dr. Sunil Paul Mathew, and all the participants of this webinar. On behalf of Department of Chemistry, St. Stephen's College, Uzhavoor, I take this opportunity to thank all the participants and who contributed directly and indirectly to the success of this webinar. I wish, first of all, I wish to express my sincere gratitude to our Bursar, Reverend Father Jins Nellikatil, for his support and throughout this webinar. And I also thank our HOD, Mr. Ebe Jimson, for the technical support. I take this opportunity to thank our chief guest, Dr. Sunil Paul Mathew. And it was a very nice topic, and it was a recent and advanced topic. So he explained the topic in a very simple way. So I take this opportunity to thank our chief guest, Dr. Sunil Paul Mathew, on behalf of St. Stephen's College, Uzhavoor. No programs can be successful without a single person. So I thank all the participants in this webinar for making this webinar a grand success. Thank you all.
Thank you, sir. So we will be sending the feedback form. You will get the certificates only if you are filling the form. So please kindly note it. Department of Chemistry, St. Stephen's College, Uzhavoor has a clear vision and mission behind each step. Today, we could take a beautiful step through Dr. Sunil Paul, Mathew, and also through the enthusiastic participants. We are so thankful to all the organizing committee members and faculties. Once again, thank you all for your cheerful presence. Reminding you about the feedback form. So let's go and shine in the science world. Thank you.